right, guys, we're going to get started. Good morning. We are going to really sort of switch gears now from, by the way, is it too dark in the back, or are you guys okay with that? You like that? Okay. Last year they didn't like that, so I just wanted to make sure. All right. Yeah, so we're going to really switch gears here this morning. We stopped talking about sort of alterations in acid-base balance and fluid and electrolyte, and now we're really moving into sort of a whole new category, and that is the whole sort of topic of immunity, and really then next week talking about sort of infection. So again, you can't talk about pathophysiology. You can't talk about things that go wrong in the body without talking about infection. I mean, as soon as you guys start clinicals when Batty's over, you're going to see very quickly that almost, all, if not most, of the patients that you're taking care of have some sort of issue with immunity or infection, or perhaps didn't come into the hospital with an infection, but maybe got a hospital-acquired infection. So we really do need to sort of spend time talking about how our body tries to protect ourselves from those sort of onslaughts. So next week we'll talk more about actual infections and the type of infections and the type of pathogens and things like that. But again, we really can't talk about infection without laying a really good groundwork and a really good sort of firm foundation today about sort of how our body tries to protect against that from even happening. So today will be sort of the groundwork, and then next week we'll be talking more about actual infections and how it affects our patients and how we treat them and so on. So today, chapters 5 and 6 are talking about the different types of immunity in our body, and a little bit we'll touch very sort of briefly on wound healing, so how our body sort of heals and how our body sort of protects itself in that way. So let's get started. Here's your objectives for you. All right, so as we move through the content today, obviously immunity, the whole topic of immunity can seem a little abstract, can seem a little hard to sort of really picture and sort of wrap your mind around when you're talking about things that are, floating in the blood or in the bloodstream or, things like that. So really as we move forward today, let's try to really, picture it happening. Try to create a picture. Try to create a movie in your head of some of these things that we're talking about. And hopefully it'll make a little bit more sense to you and not feel so quite abstract. So that being said, when we're talking about and starting with the topic of immunity, we really have to recognize that there are 3 different layers, so to speak, of immunity. So there are really 3 lines of defense, so to speak. So the first layer, the first line of defense is just sort of our innate immunity, and some other terms you might hear it referred to as natural or native. Anyone know what this first line of defense is? What's our first sort of response, or I should say protective mechanism to infection? Barriers, right. So the biggest one being skin, obviously. And there are some others that we'll talk about in just a minute. Good. So that's our first line of defense. Now, is that impenetrable? No, absolutely not. So we certainly need some other lines of defense to back that up. So the second line of defense is basically the process of inflammation. So an inflammatory response is truly our second sort of line of defense to some sort of pathogen or to some sort of infection. Now, I will tell you that inflammation or the inflammatory response is extremely nonspecific. And what I mean by nonspecific is the inflammatory process is going to act the exact same way every single time, no matter what the invasion sort of is. So it could be an inflammatory response to some type of injury, right? You slam your finger in the door. What's immediately going to happen? It's going to swell up. You're going to see sort of all those signs and symptoms of inflammation, right? So it's going to act the same if we slam our finger in the door as it is if we have some sort of bacteria that's gotten in, right? It's very nonspecific. It's just going to respond the same way every time, no matter what the problem is. As opposed to the third line of defense, which is what we call sort of the adaptive or the acquired immunity. Now, this is your more very specific kind of response. So this is now what we're talking about in terms of antibodies for specific antigens, your B cells, your T cells. They have memory so that the next time that you come into contact with this particular pathogen, the body knows how to respond that much faster, better, stronger. So it's very smart. It's very sort of sophisticated and way more specific, obviously, than the second line of defense, and certainly more specific and more sophisticated than the first line of defense. And really, the third line of defense is going to kick in when those other 2 lines fail, right? When number 1 and number 2 don't work, or they don't work enough, shall I say. So that's what we're going to start with today. Now, we'll spend the rest of the lecture, honestly, on the second line of defense and the third line of defense. But I have one and only slide designated for the first line of defense. And again, really, we're not going to spend a lot of time on it today because it's pretty straightforward. It's pretty sort of intuitive. So as we mentioned, the first line of defense is physical barriers. So skin, number one, right? But there are other things as well that certainly protect and sort of run blocker, so to speak, for our body. And that is the lining of a lot of our tracts, right? So the lining of the GI tract, the lining of our genitourinary tract, our respiratory tract. And the way that some of these barriers work is that they sort of slough off cells. So if there's anything that sort of got sort of into contact or is in the cells, they sort of slough off and regenerate cells pretty frequently. We know that just our sort of cough, gag, sneeze reflex is also a really good barrier to sort of foreign objects or foreign bodies getting into our system. Flushing, so if you get something in your eye, what happens? Your eye tears like crazy. Vomiting, same thing if something gets into your GI tract. And we know too that the mucus and the cilia in our respiratory tract also act as sort of first line barriers, first line defense for getting any sort of foreign objects or foreign pathogens into our system. And so that's pretty straightforward. It's pretty intuitive. Then we do have maybe a little bit more complicated type of barrier, and that is these epithelial cell-derived chemical barriers. So you may not necessarily always think about it, but there actually are some chemicals, some actual sort of substances in things like saliva, in our tears, in earwax, in our sweat, etc., that actually sort of protect against and sort of kind of have lysosomal action against some of these foreign bodies that might come into our system. So again, those are more sort of chemical barriers. And then obviously the normal bacteria in our gut, so the normal bacterial flora, also is a really good protective mechanism against outside bacteria or other, icky pathogens that might get into our GI system. That good bacteria, that good flora in our GI system can certainly protect against getting GI infections as well. So again, physical barriers, but then also a little bit more sophisticated chemical barriers consist of that first line of defense. So again, the immune system really does take a multi-layer approach to protecting us and to protecting our patients, and this is just sort of that first layer. Okay. Very mechanical. That's how I want you to think about this first line of defense. It's very structural, very mechanical. All right. Any questions about first line of defense? Pretty straightforward. All right, great. So, as I mentioned, thus begins the rest of the lecture. So now we're going to talk this first half of the morning today regarding second line of defense, and then we'll finish up with third. So again, as I mentioned, the second line of defense is our true inflammatory response. It's inflammation. It's our first, now to be clear, it's our first immune response to an injury. So yes, we have that first line of defense, it's our skin, but now we're actually starting to engage the immune system. This is the first time the immune system even comes into contact with any type of, foreign object. Again, it's the non-specific immune system, and as I mentioned kind of at the beginning of the lecture, it's really caused by a variety of different things. It's going to respond the same no matter what the variety of materials may be. So maybe it will be an infection. Maybe it is some sort of virus or some sort of bacteria that causes the inflammation to start. But perhaps it's just mechanical damage. Maybe, and by mechanical damage I mean like, slamming your finger in the door and your finger blows up, those types of things. But you also see it happen with ischemia, so lack of blood flow. You can see it in nutrient deprivation, so sort of malnutrition, nutrient deprivation type things to the tissue. You see it with temperature extremes. So both hypothermia or hyperthermia, so really, high fevers or really low body temperature, and radiation. And there's some other things as well that can certainly start the sort of inflammatory response. So that being said, the next thing that we need to talk about is this inflammation, this inflammatory response has many vascular responses. Okay? And we're going to spend some time talking about that. So to be clear, to start off with, the inflammatory response is a process that occurs in vascularized tissue or cells. Your body needs a blood supply to be able to sort of mount an inflammatory response. So you're going to see the inflammation or the inflammatory response occurring in places with good blood supply. And this is why. So these are the vascular responses to second lines of defense. One, you see blood vessel dilation in an area of inflammation. Why might that be? Why would it be important in an area where you're trying to sort of fight off infection or fight off foreign objects? Why would blood vessel dilation be helpful? Mm-hmm. Exactly. Right. Right, exactly. You know, it's football season, so bring in your linebackers, right? Everybody rushing in, right, exactly. So if you have a larger sort of vessel circumference, you're going to get more blood there, right? You're going to get more blood to that area. So blood vessel dilation is super important. Good. How about why increased vascular permeability and leakage of that fluid in the vessels out into that interstitial space? And we talked about this. We talked about this in our previous unit prior to exam 1 about how vessels can become leaky and fluid leaks out into that interstitial space. Why would that be important? Why is that helpful in an inflammatory response? If our vessels become leaky. And I will honestly tell you, the vessels become leaky because there's actual sort of contraction of those endothelial cells that make the vessel. So as the cells kind of contract on each other, it creates pores. It creates holes in the vessel itself that allows for the vessel to sort of become porous and for that fluid to leak out into the interstitial space. Again, why is that helpful? Anybody know? Yeah, take a stab. Exactly. So if the infection is in my tissue, and yes, my blood vessels dilate and I get all that blood kind of in that localized area, but my vessels don't allow for that good stuff like the white blood cells, the other parts of the blood that are sort of immune responders, what good is it if it just sits there and doesn't leak out into the tissue where that infection is? That's perfect. That's very good. And I couldn't have said it better myself. Very good. Very, very good. So it allows for the fluid to actually get out to the infection or to the injury. And what might that cause? What might you see in inflammation to that tissue? Edema. That's exactly why you see swelling. That's exactly why you see edema and inflammation, because that fluid has now leaked out of the vessels into that infected or potentially infected tissue. Excellent. Very good. And also then what you'll also see is the white blood cell adherence to the inner walls of the vessels, because then once they adhere to the inner vessels of the wall, inner walls of the vessel, sorry, then once those pores do open up, then that makes it that much easier for those white blood cells to just leak right through those cell walls into that tissue where they're needed to respond. Very good. Again, so vascular responses are super important to inflammation sort of happening, even being successful in the first place. And that's because you increase blood flow to the area. Also, some blood vessel dilation, actually, because you do allow for more blood to get to the area, it can actually kind of dilute the toxins in that area. So if you bring more fluid and more water and more blood to that area, it sort of waters down some of those toxins that are there present in the first place. And provides, obviously, like we said, essential immune cells out to those tissues. So perfect. Very good. And so because of that, because of those vascular responses, you see what you see in inflammatory responses. You see redness. Again, you see redness because you've got more blood flow to the area, right? It's going to have more blood, so therefore it's going to look more red, more pink, more sort of angry, so to speak. Heat, again, because you're bringing more, cells and more blood to that area. Swelling because of the edema that we talked about. And obviously pain. One reason that you see pain and the local manifestation of inflammation is because of prostaglandins. You might have heard that term before. Those really do impact and affect the nerve endings in the area, and that's why you see pain with inflammation. Okay. So again, to sum up this slide, inflammation occurs in tissue with blood supply. I hope that's sort of obvious why that's important now. It's activated very rapidly. Again, the minute you slam your finger in the door, boom, your finger swells up, right? It's rapid. It encases sort of activity of cellular and chemical components, and it's nonspecific, as I mentioned. Okay? All right. So why? What's the sort of goal? What's the sort of purpose of these things happening in inflammation? Well, it limits and controls the inflammatory process, right? It prevents and limits infection and or further damage of an infection to that tissue. It initiates that sort of adaptive immune response. Now remember, adaptive immunity is, or that acquired immunity is what we're talking about with like B cells and T cells. So it does have sort of a link. It does kind of talk to that third line of defense and let the third line of defense know when it needs help and when the third line of defense needs to kick in. And then it obviously initiates healing by bringing all those, immunity cells, the white blood cells, the other products of immune cells to that area. Good. And one of the true ways that it really does initiate healing to that site is by, we know that there's a lot of products in the blood like white blood cells and phagocytes and things like that that actually help with the removal of debris and removal of that nasty stuff, so to speak, products of inflammation, dead blood cells, products of inflammation. It actually kind of helps clean up and remove some of that cellular debris as well, and that's another really good reason of why it sort of initiates healing. Cleans up and removes some of the nasty stuff and cleans up the area, so to speak. All right. So, that being said, here is sort of a good visual of some of the things that we just talked about. We have the acute inflammatory response. Here's a good picture of some of the things we talked about. So here's the vasodilation, so the vessel kind of blows out, it gets bigger, it gets wider in that particular area, which causes the redness and the warmth. We see here the pores occurring in the actual endothelium, in the lining of the vessels, that causes the vascular permeability that allows for fluid to leak out and cause swelling or edema. We also talked about pain, so stimulation of the nerve endings by those prostaglandins that actually causes the discomfort and inflammation. But we didn't really talk about cellular infiltration or thrombosis, so clots to that area. So cellular infiltration is sort of a sign of a bacterial infection. So this would really occur with some sort of bacteria. And the reason for that is if you see any kind of drainage, so the sort of non-clinical term for drainage, sort of drainage that you see at an infected site is pus, right? That's not really a super clinical term, even though it's funny that the book sort of refers to it as that. The word we like to use clinically is exudate. Thank you very much. So you're really going to see exudate or pus at an area where there's a bacterial infection. Anyone know why specifically you see that only really in bacteria-invaded infections? Anyone know why? Mm-hmm.
The phagocytes that come through that can't fit, and so they have to come through and collaborate. But really, it's the eosinophils the neutrophils that are first that create that coloration.
Right, exactly. And even more, that's a very great specific answer. And even more general than that is, bacteria often require or often sort of elicit a white blood cell response. And what you're seeing, what pus is or what exudate is basically dead, broken up, whatever, white blood cells that have come to that area, sort of the leftovers or the byproducts of a white blood cell response. And white blood cells, as was mentioned, like, neutrophils and phagocytes and all of that are white blood cells. And that's why you see what you see at that site. Good. Very good. And then how about thrombosis? Clots. Why would that be helpful? Clots are not always a bad thing. Why is it a good inflammatory response? Why would a clot or a thrombosis at that site be good and helpful? Especially in the site of an injury, right? Why? Right, to keep it from bleeding. Very good. Very good. And the other thing is, too, is that when you have sort of that blood clot with the sort of the meshwork and the fibrous type of sort of nature that a clot is, it kind of holds that infection in that one localized spot and prevents it from sort of spreading and going somewhere else. It's sort of like a net that catches it at that site and prevents infection from spreading. So it stops the bleeding and it provides sort of a net to catch further infection from spreading out. Good. Very good. All right. So now, as you see these things on the right side of the screen, you can kind of see, pathophysiologically speaking, what causes them. And the three things, the three processes that cause some of these responses that we've already talked about are these three right here. Mast cell degranulation, and we'll talk about these in just a second, but mast cell degranulation, activation of plasma systems, and release of cellular products. And again, all three of these sort of processes are going to occur either because of cellular injury or pathogenic invasion, okay? Now let's talk a little bit about what these three are. We're going to spend some time talking about each one of these. Okay, let's begin. All right. First thing, we're going to start with this activation of the plasma protein systems. Now, this is one of these slides that I was talking about when we first started this morning where it's kind of hard to wrap your brain around. It's kind of abstract. So create whatever sort of picture, whatever sort of little movie in your mind that might help you a little bit with making this come alive a little bit more. Okay. So when foreign substances invade the body, these proteins, these are specific sort of plasma proteins in the blood, will become activated. Now, we talked about one really specific, really important plasma protein in last unit. What was that? Albumin, right. And we know how important and what albumin, what the function of albumin sort of does. Put it aside. We're not talking about albumin today. We are talking about a completely different other class of plasma proteins. So plasma proteins do a lot of different things. We talked about that one specific thing that albumin does. Now we're going to talk about some plasma proteins that work in the immune response, in the inflammatory response. And the protein systems are, again, kind of abstract here, but hang with me, the complement system, the clotting system, and the kinin system. And we'll talk about each one of these more specifically. But again, they're all going to get activated by foreign invaders. Now, all three of these protein systems contain inactive enzymes or proenzymes. And they all have to they're all sort of these cascades. And we're not going to get really specific into the sort of steps of cascades today. But what I sort of want you to understand is that, one enzyme needs to activate the next, which needs to activate the next, which needs to activate the next. Sort of the substrate of each activated, part of that cascade becomes sort of the next component in the series. That's all you need to know. We are not going to get super specific about that. What I want you to understand is that these systems are kind of like a cascade. Like one step needs to happen before the next. And that's really all I want you to understand about that. And again, albumin is not part of this. That's what I want you to understand about this slide. So let's talk about some of these systems more specifically. All right. The complement system. The complement system is a group of plasma proteins in the blood that can specifically destroy pathogens directly. Okay? It either can activate or collaborate with all the other components of the inflammatory response to start sort of an attack, to start sort of degradation of those pathogens, of those foreign invaders. And what's interesting, too, is that the factors that are produced during activation of the complement system are among truly the body's most potent defenders. So it's really important that the complement system gets activated. And again, it simply gets activated by these pathogens being present, by being there. Okay, I know, abstract. But let me give you a couple of specific examples of some of these different ways that the complement system can work. The first specific example that I can hopefully sort of make it come more clear for you is something called a membrane attack complex. Okay, it's part of this complement system. And what I want you to know about the membrane attack complex is that it's 5 sort of complement systems that join together, so they sort of all come together and work together. And how it works is that these membrane attack complexes embed themselves in the plasma membranes of bacteria. So they actually sort of latch onto bacteria and get into their cell wall, into their plasma membrane. This then, because they attack the walls of the cells, create openings, and it allows for water to rush in, fluid really, to rush into those bacterial cells. So water just keeps rushing in, rushing in, rushing in. The cells, the bacterial cells just keep swelling, 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 swelling. Well, what do you think is going to eventually happen? Burst and die. Exactly. So that is one specific, clear example that I can give you of how the complement system can actually protect and actually invade pathogens, okay? And this particular one is by attacking their cell wall and basically allowing water to rush in and burst and die. Another really clear example of the complement system is something called opsonins. Now, opsonins are basically a sort of substance that coats the surface of a bacteria. So it doesn't embed in the cell wall. It just coats the surface of it and makes it more likely or increases its susceptibility for phagocytosis, right? So once it sort of coats the bacteria, it increases the likelihood and increases that bacteria's susceptibility to get eaten by the white blood cells, by the phagocytes, okay? So that's another clear, hopefully specific example I can give you of the complement system. Does that make those that come alive a little bit more for you and give you some specific examples? Okay. But again, it's more complex than that, but that's the specific examples I want you to understand. The complement process involves a lot. So it involves, involves, approximately like 20 different types of plasma proteins that really enhance the action of antibodies. Now, what did I just say? It enhances the action of antibodies. So do you think that the complement system, even though it's part of that second line of defense, do you think it has a pretty close relationship and close communication with the third line of defense? Yes, absolutely. So it enhances the ability for that third line of defense to come in and really create a good, sort of mount a good response to that pathogen. Yeah, question? Opsonins? Mm-hmm.
Do they just increase susceptibility, or do they also act as a marker?
Well, it's kind of a combination of both. It kind of is like not really a receptor on the outside of that cell, but almost like a flag that kind of and that's why it increases its susceptibility, because it sort of makes that cell more obvious to the third line of defense, to those antibodies to come and find it. Yeah, so kind of hand in hand. Yeah, good question. Any questions about the complement system? Hope that makes it a little bit more clear for you. And that's really all you need to know about that. The second one, the clotting or the coagulation system. Okay, so again, it forms this fibrinous meshwork at an injured site or an inflamed site, right? We talked already about the positives and so why that's important. So we said that it prevents the spread of infection because it kind of creates a net or a cap over that area of infection. It keeps microorganisms and foreign bodies at the site of greatest inflammatory cell activity. So that kind of makes sense too. So not only does it keep the infection localized instead of spreading, but we know now, because of what we talked about already, that all this good stuff comes to the site, right? White blood cells, all these other sort of immune cells come to that site and leak out into the tissue. What good would that do if all of this good stuff, this helpful stuff, comes to the site and the bacteria or whatever we're talking about floats away and spreads away somewhere else where that good inflammatory cells are not, right? So it also keeps it there at the site of the greatest amount of inflammatory cell activity. We mentioned right here that a clot that forms also stops the bleeding and provides a framework for repair and healing of that tissue as well. So it kind of creates a scaffolding or a structure for that vessel to become healed as well. And the main substance that does this, the sort of leader of this and why this is so effective, is an insoluble protein called fibrin, right? A fibrinous clot. And fibrin or fibrin, sometimes you hear it referred to, is what's responsible truly for that clotting, that effective clotting or coagulation system. Any questions about the plasma protein system of clotting, coagulation system? Okay. And then the third one is the kinin system. Okay. I feel like of all the three plasma protein systems, the kinin system is probably the most sort of abstract. So let's spend a little bit of time talking about that. Kinin interacts very closely with the last system that we just talked about, the clotting system. It functions to activate and to assist those inflammatory cells that get to the area. The primary kinin, just sort of like a little FYI, just so you know this information, is bradykinin in case you ever hear that term. And it causes dilation of blood vessels and smooth muscle contraction. Again, why is that important? Why would the kinin system, its sort of process of creating this sort of vasodilation and smooth muscle contraction, why is that important again? Tell me. Yes, right. So allow for that permeability of the vessels, for that fluid to leak out into the tissues. Good, again. So that's why that particular purpose or that particular function of the Kynan system is so important, because it allows for that vessel permeability. Good. Good, good, good. So that's kind of what's most important about the Kynan system, as opposed to the clotting system that we just talked about. Really, one of the major functions of that is to keep the good stuff there with the trapping of the sort of the mesh net, and keep the bad stuff out. Okay, so that's the difference truly between the clotting system and the Kynan system. Okay. Let's talk a little bit about the cellular components of inflammation. So we talked about the plasma protein systems. That was the first sort of one from that 3 things on that diagram that I showed you. Now let's talk about a second, the cellular components of inflammation. So we have cell surface receptors and chemical mediators. So let's talk a little bit about the difference between those two. The cellular components of inflammation truly do respond to molecules produced at sites of cellular damage. So that's kind of how the cellular components of inflammation get activated. And as we mentioned already, inflammation is a process in vascular tissue. We said that already. So the cellular components of the response can be found obviously in the blood or tissues that are sort of right there surrounding the vessels, right there surrounding the blood. And these things are, as I mentioned, cell surface receptors and chemical mediators. So, cell surface receptors. Again, I'm not going to have you get super specific and super technical about this. Again, I'm not saying that these things are not important. I'm just saying for the purposes of this course and for the purposes of your knowledge as a nurse, you don't have to get super specific science-y about this. I just want you to understand the big, broad picture of this so that you kind of understand what's happening in your patients with an infection or with some type of inflammatory response. So I'm not going to get super specific about cell surface receptors. What I am going to do is give you an example. Again, try to give you an example to make it a little bit less abstract and a little bit more clear for you. So one type of cell surface receptor are pattern recognition receptors, and one specific form of that is toll-like receptors. Let's talk a little bit about what that is. They do a couple of things. One, they recognize 2 patterns, okay? So one, these are molecules that are expressed, they recognize molecules that are expressed by infectious agents, and they recognize products of cellular damage, okay? So they recognize molecules that are expressed by infectious agents and products of cellular damage. That's what cell surface receptors do. Now, the other thing that's important to understand when talking about cellular components of inflammation are chemical mediators. And I will honestly tell you, I think these are a little bit, if I had to pick, a little bit more important, so to speak. And these are the chemical mediators. Now, again, we talked about cytokines last unit. And we talked about how some cytokines are responsible for cell growth and turning on and turning off cellular growth. Again, this is a different type of cytokine, to be clear. What these cytokines do, these sort of work as messengers in the inflammatory response. And what they sort of do is they sort of go from site to site of, let's say there's an area of infection. They kind of go from cell to cell or site to site within that inflammatory response and kind of let people know, let the cells know that, like, hey, there's this infection here. We need to do something about it. They talk. They're the chemical messengers in that area to sort of organize and kind of get in line what needs to happen during an inflammatory response. So let's talk a little bit about some specific cytokines to make that a little bit more clear for you. All right. 2 really common chemical mediators or cytokines are interleukins and interferon. Okay, so this is going to hopefully clear this up for you a little bit. Interleukins is a type of cytokine that's produced primarily by macrophages and by lymphocytes. And what do I mean by lymphocytes? Give me a synonym for lymphocytes. White blood cells. Good. So interleukins are primarily produced by white blood cells in response to a pathogen or stimulation by other products of inflammation. Okay, good. And that's what I want you to know about interleukins, right? They are produced by white blood cells to kind of help stimulate and help organize some of the things that are occurring at that inflammatory site. Okay, so that's very white blood cell driven. Now let's talk a little bit about interferon. Interferons are small proteins that are released from the cells that are infected by viruses. So they actually bind to the receptors on the plasma membrane of non-infected cells, and they trigger a synthesis of enzymes that actually inhibits viral replication. So the way that I sort of think about this is I think of interferons as sort of the drama queens of the inflammation process, right? They are released from infected, so a cell that becomes infected by a virus releases interferons, and it's like, oh, I've been struck, go save yourselves, right? So it actually helps prevent viral replication from occurring from cell to cell to cell to cell, because a cell that's already infected by a virus, it's basically like, okay guys, go save yourselves, I'm no good, right? I'm done with, go save yourselves. So that's kind of what happens, is it prevents, it's a cell sort of response out to the other cells to say, I've been hit by a virus, and actually prevents replication of the virus from other sort of friend cells in the area. It's such a drama queen. All right. So like I said, it's sort of the way I think of it is it's a dying cell's attempt to save their friends, to save their other cells. So that's sort of the way I think about it. And that's how I want you to understand interferon. So interferon is more sort of virally associated. Interleukins are more bacterial and white blood cell associated. Question? Yes.
What do you mean by trigger synthesis of enzyme? Is it tagging them so that the virus can't get into the cell, or is the virus getting in but not allowed?
So viruses really have to go into cells to replicate. They can't replicate outside of cells. So it does kind of block them even from getting into those other cells. And if they can't get into the other cells, then they can't replicate. Does that make sense? Because viruses have to hide in cells to replicate. They can't replicate outside of the cell. Good question. Okay. All right. So again, the majority of cytokines are sort of classified as interleukins or interferons. Many different kinds of cells must cooperate during an effective immune response, and these cytokines kind of help that to happen, as I sort of said. This is achieved through these cytokines. They sort of diffuse over short distances and bind to appropriate target cells and affect the function of those target cells, as we just mentioned, either in a bacterial process with interleukins or more of a viral process with interferons. Does that help clear up a little bit? Maybe a smidge? Okay. Again, I know it's kind of hard. It's kind of abstract stuff. Okay. Again, and this kind of, Taylor, is another sort of answer to your question here about the action of interferon. So the viruses are in here in this cell that's infected with the virus, okay? These interferon molecules sort of bind to uninfected cells and sort of induces a synthesis of this antiviral protein, so that then when the virus tries to go to the next cell, the protein sort of blocks the sort of synthesis of the virus and blocks it from getting in. Does that answer your question? Okay. Again, a dying cell's attempt to protect other cells. So dramatic. All right. Now, the last thing we have to talk about from that picture that I had of the three different sort of processes that creates this inflammatory response, this is the third one, if you remember, and that is mast cells. Now, again, I want you... Hello? Someone's knocking at my chamber door. I have no idea what that is. We'll just keep going. So... third thing, mast cells. I really want you to understand this, okay? This is the hallmark. This is the hallmark of inflammation, is what happens and because of mast cell products, okay? So mast cells are these little cellular bags of granules, of goodies, right, that are located in the loose sort of connective tissue close to the blood vessels. So I really want you to think about them as sort of attached to or very close to blood vessels, okay? Now, when a mast cell comes into contact with the inflammation process, so the inflammatory process, what it does is it actually releases these little goodies, these little granules inside its bag into the blood vessels or into the tissue. And you really see these cellular bags of goodies, these mast cells, in the skin, in the vessels, shall I say, in the skin, in the digestive lining, and in the respiratory tract. And the two main components of what sort of makes these sort of consist of these little granules inside this bag, inside this mast cell, are, and again, I want you to remember this. This is something that I want you to forever remember now about the inflammatory response. Mast cells contain histamine and chemotaxic factors. Let's talk about what those are and make that clear to you. Okay. So, what do you know about histamine? What does anyone know what histamine does? What do you think? Shout it out. Okay. We think about histamine when we think about allergies, right? But why? What does histamine really do? Anyone know what sort of the function of histamine is? Yeah. Yes. So histamine is one of the main reasons why you have that endothelial contraction of the vessels that allows those pores to form, that allows for fluid to leak out. So, and it also really helps with vasodilation. So again, hopefully now it's starting to come together for you that histamine is, it's obvious why this might be one of the hallmarks of inflammation, and histamine creates that vasodilation. It's what actually causes that vasodilation in the inflammatory response, and it's what causes that fluid to leak out of those pores in the vessels out into the tissue. So that's exactly why we think of histamine when we think of an allergic reaction, because allergic reaction is kind of an inflammatory response as well, sort of a hypersensitive inflammatory response. So that's why, because we know histamine now causes the vasodilation and the swelling. That's why, like in an allergic reaction, you see the swollen lips and the swollen tongue and their eyes are swollen shut. Why? If histamine's been activated, why do you see the big lips? They're worried about their airway closing off because of the swelling. The eyes are sort of swollen shut. Why do you see that now? Put the pieces together. Why do you see that swelling in someone's face like that, and in an inflammatory response like that, where histamine's activated? It's swelling, right. Histamine has allowed for all that edema to occur in that site. Perfect. Very good. You're starting to put the pieces together. And what also might you see if histamine is truly, truly activated? What might you see hemodynamically? And by hemodynamically, I mean like to their blood pressure. What might happen to someone where histamine's been really activated and you've got this so profound vasodilation response in the body? What might you see hemodynamically speaking to these people? Hypotension, low blood pressure. And again, the reason for that is we talked about that vasodilation of the vessel. The reason you see low blood pressure is because here's the vessel. It's nice and filled with a lot of volume, with a lot of blood. And now histamine's kicked in and it blows out the vessel. It vasodilates the vessel. The blood volume hasn't changed, but what has happened proportionally now to the tank? The tank's gotten a lot bigger, right? And blood has not been added. So in terms of sort of the proportion of the blood in the tank, it bottoms out, right? You have this tank, now you have this tank, and the blood goes boom, and your blood pressure bottoms out, right? So you also see, with a profound histamine response, you see pretty profound hypotension at times as well with our patients. Good. Okay. Now, chemotaxic factors. Everyone clear on histamine? I want you to be super, super clear about histamine. Yes? Okay. Chemotaxic factors. Chemotaxic factors help with immune cellular movement. So we've talked about how important it is for those immune cells to get to the site of invasion. Chemotaxic factors help them do that. It sort of guides them and leads them to those areas where the inflammatory response is needed. So particularly, it helps with the movement of white blood cells, of getting white blood cells to that area, to that site of invasion. Okay? And you see here at the bottom of the screen sort of the processes that activate mast cells and chemotaxic factors. Physical injury, chemical agents, immunological processes, and actually the action and the function of some of those toll-like receptors that we just talked about also help with mast cell sort of release of these goodies, which are histamine and chemotaxic factors. Okay, and the way that it happens is what we call degranulation. And basically what degranulation means is that the mast cell just sort of opens up and releases these granules, releases these contents, which are histamine and chemotaxic factors. Okay? All right. This is kind of a review of what we just talked about in terms of histamine. So hopefully this slide makes sense to you. It's a vasoactive chemical mediator that causes smooth muscle contraction and increased permeabilities of those capillaries to allow for white blood cells and other things and other fluid to leak out. And that's why you see the swelling and the vasodilation. Okay? And again, that's also why you see those big swollen lips, their eyes are swollen shut, their airway is getting tight and swollen shut in an allergic reaction, because that's also an inflammatory response. So if you're ever curious or you forget the actions of histamine, just think what someone's face looks like in an allergic reaction, and you'll remember quite quickly what histamine does. Okay? Now, I don't want you to really pay much attention to this right side of the screen, but I want you to remember what the process of degranulation, so those granules coming out of the mast cells, and what it actually causes, right? So histamine causes those vascular effects. Chemotactic factors cause those sort of movement of white blood cells to the area. Okay? And this is, again, just kind of a review of the chemotactic factors that we talked about. All right. Any questions so far? Any questions about what we've talked about up until this point? Okay. Now, before we move into talking about sort of the third line of defense, we really can't leave this topic yet until we talk a little bit about the actual phagocytes. Okay? Now, phagocytes are white blood cells. And there's, as you know already, there's a lot of different types or classes or families, so to speak, of white blood cells. Let's talk a little bit about some of the white blood cells that you might see at this site of inflammation in this second line of defense. Okay? The first one, so these are sort of key players in the second line of defense and in inflammation. The first one here, as you see with stars, are neutrophils. Why might I have starred this? Why might I have starred neutrophils and put them first in the list that I'm talking about? Yeah. They are. They are the predominant phagocyte in early inflammation, in the inflammatory site. And you see here what I have on the screen. About 6 to 12 hours after some type of injury or some type of inflammatory response, you'll see the neutrophils there. They're responsible for removal of debris and dead cells and for the actual destruction of bacteria. Good. Now the next one, I also have stars by, macrophages. Why might I have stars by that one, number 2? They're the second. I'm not trying to trick you. They are the second responder, so to speak. They typically arrive at the inflammatory site about 24 hours or even maybe a little bit later after the neutrophils are there. But number 1 responder, number 2 responder are neutrophils and macrophages respectively. Okay? Another thing that you might see in terms of phagocytes at an area are monocytes. Now monocytes are, as you see here on the screen, an immature form of white blood cells. Why might, if I would draw a blood test, if I would get a blood sample from a patient who is in an acute sort of inflammatory response, why might I see an increased level of monocytes in that blood sample? Yeah. Right, exactly. So the body has given out the alert that we need more blood cells. We need more, and here's the key, we need more mature white blood cells, like the neutrophils and the macrophages. So all the neutrophils get used up, all the macrophages get used up, and so what you see then is that you start to see these immature forms of white blood cells out floating around in the tissue or floating around in the vessels as well, because all the mature ones have been used up in this sort of acute inflammatory response. So now we're all used up. We've got to use the youngins, right? We need the immature sort of, less effective ones, so to speak, but we need you because we've used up all the neutrophils and macrophages. So then you start seeing these immature, sort of less effective white blood cells floating around out in the blood because we've called on the reserves, essentially. So you'll see an increased monocyte count in an acute inflammatory process because the neutrophils and the macrophages and the other more mature phagocytes have been used up. Does that make sense? Okay. And then we're not going to spend a lot of time with this slide, but other phagocytes, just sort of for FYI, are eosinophils. We see this type of phagocyte used a lot as a primary defense against specifically parasites. And you also might see eosinophils more sort of responding during allergic reactions. So that's kind of eosinophils. And again, it should make sense to you that you see a sort of relationship between eosinophils and allergic reactions because look right here, it says it regulates vascular mediators released from mast cells. And what are those things released from mast cells? Histamine, right? So hopefully it should make sense to you that eosinophils play a really big part in allergic reactions, and you see what you see. You see the histamine release in those allergic reactions. And then dendritic cells we're not going to necessarily worry about today. Okay. So phagocytosis now is the multi-step cellular process for these phagocytes to actually eliminate pathogens and that foreign debris at the site. Okay? Now, I kind of have a step-by-step sort of process here for you. So first you have recognition of some sort of pathogen that the phagocyte sort of notices and adheres to. So it adheres to its target. Then you actually have engulfment, so ingestion of that pathogen by the actual phagocyte. I sort of think of step 2 here as like Pac-Man. So you sort of have the phagocyte and now you've actually engulfed it by endocytosis. It takes it into the cell. Phagosome formation is just that little pocket, that little sac now inside the phagocyte that sort of holds and sort of encases that pathogen. And then you have fusion with lysosomal granules within the phagocyte that sort of degrades and breaks up that pathogen. And then, voila, number 5, destruction of the target. Now, I could sort of, talk until I'm blue in the face about these steps, 1, 2, 3, 4, 5. I've included a link here for you to YouTube. It's, not super exciting or super thrilling, but it's a really good sort of visualization of these 1 through 5 steps. So I'm not going to actually play it for you right now, but I've included it for you just for your own reference and so for you to refer to as you're studying and looking. If these 5 steps are not clear to you, the visualization there on YouTube makes it super crystal clear. Okay, so that's there for you for your reference and referral. Any questions about phagocytosis? It's kind of a review for you, I know. Okay, all right. So now let's talk about, more clinically speaking, what we see in acute inflammation. Okay? We've talked about sort of the chemical and cellular responses to inflammation that occurs in our patient's body. Very sort of abstract, very hard to sort of wrap your head around some of these things. So now let's move into the real world, right? Enough with that abstract, stuff. And now let's talk about what we actually see, what we see in our patients. And you can kind of divide inflammation into acute and into chronic inflammation. So let's talk first about acute inflammation. So we know that, if you think about a site in your body that's inflamed, right, we know that it's usually pretty self-limiting. It kind of takes care of itself, runs its course, and then no longer do you have inflammation. So you're going to see during this self-limiting sort of acute type of inflammation, you're going to see those local manifestations that result from those vascular changes that we've talked about already. So corresponding to that leakage of fluid out into the tissue, you're going to see the heat, you're going to see the swelling, you're going to see the redness, again, because of that vasodilation and that leakage of fluid. You're going to see pain because of the prostaglandins that we mentioned. And you're going to see those exudative fluids, or that pus. Or wound drainage to some of those sites. Again, that's all considered acute. If you see this type of fluids and type of drainage, that is acute inflammation. And to be clear, acute inflammation really only continues until the threat to the host is eliminated. So that's sort of the whole purpose, if you will, of acute inflammation, is just to sort of protect the host while there's a threat going on. So really, acute inflammation, the self-limiting time frame that you're talking about that is considered acute inflammation is really 8 to 10 days from onset to healing. So it's quick. That's acute. That's acute inflammation, right? And that's when you're seeing the drainage and you're seeing that redness and that warmth at the area, etc. So let's talk a little bit. Again, we can't talk about our patients and sort of the clinical picture of inflammation without talking about the delicious topic of exudative fluids. So what kind of fluids might you see at areas of inflammation? Okay, so the first type of fluid that you may see with your patient is serous exudate. So this is more of your watery exudate. It kind of has that straw, kind of yellowish look to it. It really does sort of indicate early inflammation, so you'll see this very early on in an inflammatory response. Give me an example. Give me an example of some type of threat to the tissue or threat to something where you might see this watery, clear, yellow straw-colored fluid. Yeah. Blister. Perfect. Very good. That is the perfect sort of example of a serous exudate that you find at a site of inflammation. A blister is certainly inflamed, right? It's a process of inflammation, and that's the exact type of fluid that you might see there. How about fibrinous exudate? So this is more of your thick, clotted exudate. It oftentimes does indicate, although still acute, it indicates more of an advanced inflammatory response. The example that I can give you for this is like the kind of exudate that you might find in the lungs of someone with pneumonia. So again, clinically speaking, as a nurse taking care of a patient, if someone with exudate, they have pneumonia, you might see this kind of exudate come up when they cough. They might have a really loose kind of junky cough, and they actually can sort of spit, sort of expectorate out some of this exudate. And it's that really thick kind of, green clotted type of exudate. That is fibrinous exudate. So again, trying to put a clinical, more real-life spin on that for you. Another example of exudate that you see at the bedside, clinically speaking, is what we call purulent exudate. Anyone heard that term so far? Okay, good. Maybe in Batty, is that where you, okay. This is what we call in the non-sort of clinical term pus, right? And again, what does pus or purulent exudate indicate? Bacterial, specifically bacterial infection. Okay, so I also have a really good link here on YouTube of someone, so this is the kind of exudate that you might see in someone with like a cyst or an abscess. And I have a really good video here for you on YouTube of someone getting I&D'd. That term is incision and drainage, where there's so much fluid and sort of purulent exudate in a very sort of walled off capsule that sort of is an abscess, that they actually have to make an incision and actually drain it, actually sort of let that drainage, let that purulent exudate come out. Okay, this video on YouTube is fantastic. If you're a dark, sort of strange ED nurse soul like I am. I mean, you can just see the pressure built up in this abscess. As soon as they make an incision, it just starts leaking out. You can see all of this sort of purulent exudate. But I guess it's, and I showed it last year in class, I guess it's so graphic that now it's blocking me on this computer, so I can't show it to you today. But if you'd like to go on your own computer and go look, take a look. But again, it's the perfect sort of visual of purulent exudate. Again, usually because of a bacterial infection. The one on this YouTube video that I'm showing you is specifically because of a staph infection in the armpit. So it's a perfect example of that. Yeah? In the ED, no. So like in most cases, no. It would be a physician actually doing the incision and the drainage, but we're there for pain control. We're there for other sort of things as part of the process. So you can certainly see this in the process. Support, emotional support for the patient. And then last but not least, hemorrhagic exudate. So in combination with the purulent exudate, you also might see some bleeding. Some actual sort of blood in this type of exudate, and again, just it indicates bleeding and some sort of inflammatory processes in that area. So you also might see a little bit of blood in that purulent exudate as well. And I'm so sorry I can't show it to you, but you can go look on it for yourself. Okay. How about more systemic? So those are sort of the local manifestations of acute inflammation. How about more systemic manifestations? So we see fever, right? We see hyperthermia a lot with infections. And why? We think of fever as a bad thing, But really, fever is very, very helpful in inflammatory responses. It truly, especially when talking about a bacterial infection, it truly turns up the heat. It really sort of turns up the heat on bacteria. Bacteria do not like a hot environment. They do not like a really hot situation. So fever can actually help by sort of creating this unpleasant environment for bacterial growth. So it helps with bacterial, sort of stopping bacterial growth. It also increases the metabolism of the patient. So our patients that have a fever, it actually revs up their metabolism, which also helps facilitate healing and helps facilitate phagocytosis. So fever is a good thing. And it can be sort of triggered by exogenous and endogenous pyrogens, which are sort of substances that are sort of released from the bacteria that just so happen to activate our hypothalamus and crank up the body temperature, which then sort of helps diminish the bacteria. And then the other thing we really do see in sort of acute inflammation is leukocytosis. That truly is a term that just means increased number of circulating white blood cells. So if you see the white blood cell count jump up in our patients, when we're doing, looking at their lab values in the clinical setting, if you see a white blood cell count jumping up, you know that there really is some type of acute inflammation, acute inflammatory process going on. So again, all more systemic manifestations of acute inflammation. And then a couple more here. You'll see something called, you'll see some of those plasma proteins that we talked about get elevated as well. So you can actually see an increased plasma protein synthesis. And again, if drawing the blood, if drawing, getting labs for a patient, you can actually see some of these plasma protein systems, their level in the blood is actually increased. One specific one is called C-reactive protein. We use this a lot in the emergency room setting. If the C-reactive protein is elevated, you know it's a pretty good sign that there's an acute inflammation going on. Don't know where exactly. You have to do some more investigation to find out where the inflammation is occurring. But if it's elevated, it helps you sort of hone in on, yes, this is an acute inflammation. And then another plasma protein that's elevated in acute inflammation is fibrinogen. And here's just sort of another real-life clinical example here for you on the screen. So we know that fibrinogen kind of helps with erythrocyte red blood cell adhesion. It kind of makes the red blood cells more sticky because it helps, again, keep them in that site of inflammation like we talked about earlier. So we know that increase in fibrinogen leads to increased erythrocyte adhesion. But clinically, if you, again, draw someone's blood, take it to the lab, and centrifuge it, if the red blood cells are more sticky and they're sticking to each other, they're heavier, right? Because there's more of them clumped together. So if you centrifuge that sample of blood, the erythrocytes are going to sediment out. They're going to drop quicker in that centrifuged sample of blood because they're heavier, because they're all stuck together because of this increased fibrin. So if you have something called an increased sedimentation rate, it obviously is a sign then of acute inflammatory response. There's an acute inflammation process going on because the red blood cells have dropped faster because they're heavier because of this increased presence of fibrin. So that's also a really helpful clinical example, clinical sort of lab value that will show you if someone is in sort of the process of acute inflammation or not. Does that make sense? We do that a lot in the emergency room, and you'll hear about that as you get more into your clinical experiences. And just sort of for your reference, it's usually called, that you'll hear someone say an increased sed rate or an ESR, right? Again, increased sedimentation rate of those red blood cells. Again, good, clear sign of acute inflammation. You'll hear about it in clinical, I promise, and you'll be like, ah, I know what that is. I know what that means. I love it when it starts coming together. Okay. And then last but not least, chronic inflammation. So what did I say? Acute inflammation is sort of, it's like, what did I say? Yeah, right, 8 to 10 days-ish. Chronic inflammation is anything sort of lasting 2 weeks or longer. And it's usually related to an unsuccessful acute inflammatory response. So something happened in that second line of defense. The inflammatory response did not work correctly, did not work properly, did not mount a good enough response to sort of take care of the situation, and now we're more into a chronic inflammatory situation. Characteristics of a chronic inflammatory response is you'll see more dense infiltration of lymphocytes and macrophages at that area. And you can actually see something called a granuloma form. Now, a granuloma is just this mass containing macrophages that forms there at that site as a result of inflammation and sort of isolation of that infected area. So again, when you see things like that redness, that warmth, that pain, that tenderness, these lab values that I just mentioned, that's acute. You're not really going to see a lot of those sort of manifestations in more chronic inflammation that's lasting longer than 2 weeks. Okay? All right, I know I'm kind of running over where I normally stop for break. Let me just do a couple of quick things here and then we'll take a break, I promise. I didn't forget about you. Okay, last thing we're going to talk about before break. And again, you kind of have to lump this into this conversation of inflammation and this second line of defense. And that is this topic of resolution and repair. So I mentioned we're going to talk a little bit about wound healing, and here it is. Okay? So there's two different types of wound healing. Either resolution or repair. Let's talk about what those are. So resolution is basically the returning of an injured tissue to the original structure and to its original function. Okay, that may not make a lot of sense to you at the moment, but let's talk about repair now, and then we'll kind of go in more depth to both of these. Repair, a wound that is sort of healing by repair is replacement of destroyed tissue with scar tissue. So the difference between repair and resolution is that I just said repair is the replacement of destroyed tissue with scar tissue. Scar tissue is not functional. It is not the normal type of tissue that should be present at that site. So it's very different than the definition of resolution. In that resolution, the injured tissue goes right back to its original structure and function. Therein lies the difference between resolution and repair. Scar tissue is not functional. It's not near as elastic. It's composed primarily of collagen, but it doesn't sort of restore to that normal sort of strength and elasticity of that tissue, as opposed to something that heals properly through resolution. Let's talk about the difference. Okay, resolution. Destroyed tissue are able to reform themselves. It depends on the type of cell whether or not resolution can occur. It also occurs when little tissue, and this is how I really want you to remember resolution. Resolution occurs when little tissue has been lost or injured. Okay? And another sort of way to think about this term resolution is something called a wound healing by primary intention. Let me give you an example. Think of a surgical incision. Okay? Nice little cut. What happens at the end of the surgery? They take those two pieces, the two sides of the wound, they put it right back together nicely, suture it up. It has a nice little scar, and because there's not a big gap in the wound, there's not a lot of scar tissue that forms, because those two sides of the wound are put right back together. We call that well approximated, when the wound comes right back together, and you have this little minimal scar. Okay? That is the perfect example of a wound healing by resolution and a wound healing by what we call primary intention. Okay? As opposed to, so primary intention, wounds that heal under conditions of very minimal tissue loss. Okay? That's primary intention and resolution. Okay? Here's a great example. Okay, so this is not a surgical incision, and this also is not a hand trauma patient that I took care of in the ER. This is my husband. About a year ago, we live in an older home in the Brookside neighborhood here in the city, and about a year ago, he was trying to open one of our windows, and they're old, sticky windows in this old home, and he was like trying to unjam this window and put his hand right through the pane of glass. This picture actually looks much better than what it actually looks like. The angle or something on there makes it look better. When I came in and found him, like this whole part of his hand was like hanging down here like this. Yeah, right through the glass, just slashed it. This part of his thumb was like this. So anyway, this is what it looked like before. And look how beautifully they just well approximated those edges right back together, sutured it up, and look how much better that looks than that, right? And so this is a perfect example. No, it's not a surgical incision, but this is a perfect example of a wound healing by primary intention, of just bringing the sides back together, very minimal tissue loss, very minimal scarring. So this happened a year ago, and as I was putting my lecture together for today, I texted my husband and was like, at work, take a picture of your hand now and send it to me, because I want to put the sort of the way your hand looks now to show you how little scarring there is. Look at that. It's beautiful. You can sort of see a little bit right there and a little bit right there. But look how beautifully that healed, without a lot of scar tissue, without a huge incision. Now, never mind the fact that this little cut that he had right here in the palm, so this one right here, actually cut his ulnar nerve. So he has numbness now in his ring finger and his pinky. He actually had nerve repair done like a few days after this happened. But you know, nerves, they're never normal again. So he's got some decreased feeling now in these two fingers. But never mind that. Look how pretty it looks. Look how well put together this primary intention healing happened. So anyway, this is a great example of primary intention healing, as opposed to a second intention, or repair, right? So repair, tissues that have sustained extensive damage or those incapable of regeneration heal by the process of repair. So this is not a well-approximated wound. An example of a sort of a wound that it would heal by repair would be like, let's say a trauma patient who had a motorcycle accident and fell off the bike, had this huge gouge out of his thigh from road rash kind of thing. Can you take that huge gouge out of someone's leg and put the sides back together and well approximate, sort of suture it up? No. So how's this wound going to heal? You can't bring the sides back together. So what happens is in primary or in secondary intention or repair, the wound heals from the bottom up and the sides in over time, right? So you can imagine a person who's got a huge gouge out of their leg from a motorcycle accident, or my husband's hand, who's going to have the bigger, worse-looking scar? The person who's got the big gouge out of the leg that you can't suture back together, right? It just has to heal very slowly from the bottom up and the sides in. That's secondary intention. Wounds that require a great deal more tissue replacement, like an open wound like that, is secondary intention. Another clinical example I can give you is like someone with a pressure ulcer. So here, this is a stage 2 pressure ulcer because it's actually open and down to like the adipose tissue under the skin. Again, can you well approximate these sides and put it together and suture it? No. So you have to let it just heal from the bottom up and the sides in. And yes, it heals, but look at that scar, right? There's a lot more scar tissue formation there than, say, again, the example of my husband's hand. That's the true sort of difference between primary and secondary intention and resolution and repair. Does that make sense? Is that clear? Okay. Now, this slide is just sort of for your FYI, the different phases of wound healing. So phase 1, phase 2, phase 3. To be clear, all of these phases will occur in the same order and in the same way, no matter if it's, primary intention or secondary intention. They still occur. The phases still occur, no matter which type of wound it is. And then here is just kind of a good sort of visual for you. So a wound healing by primary intention, aka my husband's hand, or secondary intention, aka a pressure ulcer. So you see how much bigger this area for tissue replacement is than a wound that heals by primary intention. I hope that makes sense to you and is clear. Okay, sorry for the late break. Let's take a break here. And then we'll start back up with the third level of defense.